If everybody can take their seats, we're going to start up again with a demonstration of tools for WGS analysis. Okay, I want to welcome Glenn Tillman with the Molecular Characterization Branch of OPHS at USDA FSIS, and he's going to talk about tools for WGS analysis.
Hello and good afternoon. Thank you for the invitation to come today. It's a very good pleasure. After hearing all those great talks this morning, you really set the stage well. Those first three talks went into great detail about kind of where we're going with whole genome sequencing. So the rest of the talk, so I hope I have something good for you all to add. So without further ado, that's me. Don't like the picture very much. Okay, and this is what I'm kind of planning on talking about, is short and to the point, give you a little background. But number two is the bullet, what are we doing with this data? And that's what most people are here to hear about, so we'll go straight into that. So some of these things Dr. Goldman already spoke of on behalf of our agency. Why are we using WGS? To support foodborne illness investigations, to support our mission goals as part of FSIS, and mainly, and more importantly, for the whole point of this, is alignment with public health partners. You see a lot of familiar names who've been up here speaking over the last day, and 2 days considering the NARMS it was last week. Earlier this week, excuse me. So where are we currently right now? We currently have 12 MiSeq sequencers in our laboratory system. 8 of those are in our Eastern lab in Athens, Georgia, where I'm located. 2 are in our Midwestern lab and 2 are in our Western lab. We, in fiscal year 17, really ramped up and sequenced around 7,200 isolates, so it's quite a jump up and from our capacity building perspective. We do collaborate really well with our public health partners, and we do consider epidemiological information in all that we do with this new emerging tool, as many of the speakers have alluded to this morning. It's one big package of information that you get. Finally, we do work really strongly with our NARMS partners. Dr. McDermott gave a great talk earlier on some of the tool, how this tool can help enhance what we're doing, and we're trending with antimicrobial resistance genes. Oops. So I want to point this out to you as a reference guide. Dr. Klimke gave a nice talk on NCBI. But here are some of the top bio projects that we and FSIS contribute to. Most of these are genome tracker specific projects. A few of these are set up specifically for us with our efforts with the NARMS program at FDA CVM. So many of these are from the cecal environment of our 4 major animal commodities. So please take note of those and peruse those bio projects as needed. And just this last shows a kind of trend, of upwards trend of how many isolates we've gone from kind of a low level in fiscal year 2014 to where we're at now at over 7,200 in fiscal year 17. We plan on going even further this year. We talked a lot about metadata today. Here's a nice example of one of our biosamples. We do release who collected, of course, the collection date and the state in which it's collected in the isolation source. We try and be very descriptive, as Bill was mentioning in his last talk, of how he could collapse some of those commodities into a certain product type, non-meat product swab. Okay, now on to the more of the analytical tools that we may use. I was actually asked to do a demonstration, but I couldn't bring myself to do that in command line in front of you all and then streaming over the web, so we're just going to stick with some screenshots. We'll start with how we do quality control assessment, antibiotic resistance gene detection, salmonella and STEC serotype determination, and STEC virulence characterization, and finally phylogenetic comparisons. So it'd be a lot of stuff coming at you really quickly. Currently, our group is responsible for characterizing upwards of 12,000 isolates per year, and that includes antimicrobial susceptibility testing, serotyping, PFG, and whole genome sequencing. So we're doing all these tests in parallel, so it's a pretty busy group and a lot of results coming out of there. The goal over the next couple of years is to continually develop one single workflow, because as we know, we can get all these types of information directly from whole genome sequence data, and we want to continue to try and pursue that as much as possible. So an overview. We start with the sequencer. I think this might be the first picture we've seen in the MySeq today. Again, we have 12 of these. If you can see up here, you'll see kind of what a FASTQ format looks like if you're not familiar with them. It's a different type of format than most of us were used to working with over the past 5 years. It's highly compressed, but even at compression rates, it's still around 300 megabytes for two files together for a single isolate. So you're talking large data sets. That data can goes one of three ways actually. You input the FASTQ into an assembler. In this case, I'll show a picture of the CLC Genomics workbench. We also use spades to do assemblies, de novo assemblies. We do a quality control pipeline where we assess coverage from the raw files, the average quality, and nucleotide balance. All these were worked out in our GenFS partnerships with NCBI and FDA and CDC. FASTQ files can go into the whole genome MLST and the bionumeric 7.6, which was talked about by Dr. Besser earlier. LiveSet and SNP pipeline are two types of high-quality SNP analyses that are actually publicly available, and those I have some screenshots of that later on. And then finally, the NCBI pathogen isolate browser. That one's nice. All the heavy crunching's done on the NCBI side, and we don't have to do that. Okay? So once we've got our assembly, we do another set of QC, quality control. We look at file size. That's important. 1 byte equals a base, essentially. We want to show that we want to have some level of correlation that our assembly is very correlated with actual bacterial genome size. 3 megabytes for Listeria monocytogenes base, Listeria monocytogenes assembly. That correlates nicely with 3 million bases, essentially is what the take-home on that is. We look at certain other metrics, N50, number of contigs. The lower number of the contigs, the better the assembly, the better the sequencing quality that went in. We want low number of contigs. Finally, correct organism. That's very important to us. We have multiple things in-house to assess that. On the NIH side, NCBI, Bill Klemke has talked at numerous conferences about how there still seems to be some level of sample mismatches and mix-ups. We try and avoid that all throughout our pipeline here and with our other assays by double-checking at multiple points. Now, here's the FASTA format, as you can see. Looks a little bit different than the FASTQ file. Again, you've gone from 300 megabytes to around 3 megabytes. So you've highly compressed really complex data into a much smaller, workable format. Then you're going to input that FASTA into our downstream tools. Determination of MLST, multilocus sequence type, antibiotic resistance genes, virulence profile, and serotype determination. And potentially you can even do different types of phylogenetic comparisons using MASH tree. So to start off, we'll talk a little bit about the antimicrobial resistance we use with whole genome sequencing data. That's been talked about a lot, again, over this week. We want to work to identify new genes of concern. That's been a big part of the last couple of years, and FSIS efforts have been part of that with the CTXM65 and other genes. As I mentioned, the CTXM65. We have colistin. We've worked to identify, do we have any of these sequences in any of our sequences that we're putting up on NCBI? Quinolone resistance and the spread of plasmid-mediated quinolone resistance, QNRB19. We work with partners at NARMS with that. Linazolid resistance as well. Okay, so the overall kind of workflow, you saw what a FASTQ file looks like. So it goes into the QC and assembly pipeline, and then from there, the assembly goes into our BLAST database. And the output is a resistance gene profile. So that's just a really quick run over what we're doing. But where do we get the antibiotic resistance gene database from? We use ResFinder currently. We get that from the Center for Genomic Epidemiology. And much like our partners, we update that on a really very, very frequent basis. Here's the output. Here's the black box in which we work. Most of this is done in command line, and there's a reason for that. All these tools are publicly available, and you can do them in most browser-type formats, but you can do that for 1 to 2 isolates. But when you do it for 7,000, you got to have a much, much better and more efficient way of doing it. That's where, in our hands, command line comes in. This is kind of a typical output that you'll see. And then in the end, we do a lot of formatting with our bioinformaticists. A lot of their work is in formatting, how we get that in a file that's usable for other people. So in this particular isolate, we identified 5 different genes conferring multiple levels of resistance, including BLA-CTXM65, TET genes, and some sulfonamide and aminoglycoside genes. All this can be found, again, publicly for the download portion at bitbucket.org, which is I believe can actually be linked to from the genomic epidemiology site at this point. Okay, so one of the things we want to show was that phenotype and genotype comparison that has been talked about. So we did contribute to the NCBI and FDA initiative recently to identify what that correlation was, but we did our own work in-house with the 2016 SQL NARMS data set, which is essentially 1,190 isolates. Dr. Goldman showed this earlier, and you can see and hopefully appreciate around 97 to 99% correlation. Going like everyone else has mentioned, there's some gentamicin issues, but overall this is a pretty tight correlation. Okay, Salmonella serotype determination, very similar to what you saw before. Assembly is key, putting it through our custom-made BLAST database based off of SeqZero, developed at the University of Georgia and the CDC. That one is actually available on GitHub. A lot of these different tools are available on GitHub, so you'll see really faint screenshots up here now against this backdrop, but you will see they are available, and you can use those. So to give you a little bit more background on that, several years ago when we first started this initiative, SeqZero was just coming into its own, so what we did was an exact matching algorithm using some Python scripts that we developed. So now we currently still use those Python scripts in conjunction with SeqZero, remove all the redundant factors, and use that against a dictionary to identify the serotype. So in our hands, we looked at over 4,200 isolates, and we found about a 96% rate where WGS matched that of the reported serotype result. We had about 3.8% where WGS did not match the serology result, and that typically was just incomplete genetic factors that we did not get, allowing us to call it serotype. In those cases, those are sent to NVSL to do traditional serology. Okay, this is getting a little familiar to you at this point. Just one thing I want to highlight is how important it is to do the assembly from the FASTQ files, but then also to utilize already existing databases to do your work with. The virulence typing and MLST typing that we do for STAG we find is very important. We use that from the Center for Genomic Epidemiology as well. We also use Virulence Finder and Serotype Finder, which were both mentioned earlier in previous talks. Another output for that. And here's an O26 strain. One of the nice things I like to point out, too, is previously in FSIS we didn't get the H-type. It's not part of what we used to do. But now we can get the H-type. A lot of them are H11, H7, which they may be. Sequence type is very important to us as well. It helps us, actually I'll give you an example later on of how it helps us kind of predict what maybe the O-type might be. The STX type, that was talked about earlier, we can actually look down to the STX1A in this case, and then the EAE allele beta in this case as well. This was an analysis that we just recently did right before we came. This is around I think 300 or 400 isolates, and we looked at our top 6 non-0157 and our 0157, and what did we get out of the characterizations? We looked at what are the top MLST types. So this, looking at sequence types, sequencing allows us to kind of go back anytime retrospectively and do this. Another very important point about sequencing, you can always do retrospective analyses. So you start to see that we do have, within each serogroup, we do have a predominant sequence type within each one of those. Well, one of the ones I like to bring attention to is ST11 on the E. coli 0157H7. In our hands, our commodities, our isolates, those tend to be very familiarly known as ST11. Various Shiga toxin types in combination or alone. EAE types, we tend to see gamma as the top in all of our E. coli 0157 isolates as well. And our top serotype, as you might expect, 0157H7, not unexpected. But then we do have, within our our other serogroups, we start to see 100% are H11, H8, and so forth. One of the nice things whole genome sequencing allows us to do, previously we couldn't do, was we can start to identify H types on serogroup 0157 that we normally would have said is a negative for 0157. Previously we've had some H11s and H29s. In both of those cases, neither one was EAE positive nor STX positive. Okay, and then finally for phylogenetic comparisons, we use all the pipelines that have been developed by our public partners. Many are here. Lee Katz's LiveSet, which again is on GitHub. The pathogen pipeline detection Bill talked about was a great tool. The FDA SNP pipeline also on GitHub. And then WGMLST, which is a very nice tool, bionumeric 7.6 from the CDC PulseNet, and there's a screenshot. Here's some output from the pathogen isolate browser. You saw this slide earlier from Dr. Goldman, and about where do some of our top serotypes in our commodities, where do they line up as far as within 10 SNPs of a clinical, 10 SNPs or 20 SNPs of a clinical, or just are they in a SNP cluster? And you can see these numbers, and you see some of the things like Kentucky. There's 0% within clinical. Again, this is all hinging on all the clinicals are being sequenced in real time. We're kind of ahead in that we're sequencing everything in real time, and as soon as we're caught up in the next year with every clinical going on NCBI, maybe we'll see different numbers at that point, but that's just something to consider. This was also presented by Dave Boxrud. This is some work we did with pattern 4 enteritidis, very predominant. 50 to 60% of our enteritidis isolates are pattern 4. So you can start to see, you start to see a delineation just looking at SNPs alone between those pattern 4 clusters, which are on your screen more of a reddish-orange tinge to them. You can start to see that they kind of break apart where with just PFG pattern alone, they've been clustered together. Okay. The last example, I'm almost done here. We did some work with harborage of listeria strains in some processing environments. And let's have one example of one establishment we had where we had about 14 or 15 isolates collected over a 4-year period. With this, we used all three different pipelines to show the concordance between those 3 pipelines and to look, what do we see here? So we used the SNP pipeline, LiveSet, and WGMLST, which is hidden behind the placard. We did see a very strong concordance between all 3 of those pipelines. They did cluster all the same isolates together. I defined in this one a cluster of 20 or less. One of the interesting things that you can see is there are several clades. Those are starting to break apart, these from here, which makes sense because these actually have nothing to do with any of these events in this establishment right here. So that's very, very important. The nice part is with PFG, all these had the same primary and secondary PFG patterns, and we were actually able to start breaking those apart and teasing those apart using some of the newer tools that are developed for whole genome sequencing. Okay, and finally, I've had one more slide. How else are we trying to use genotypic data? Again, this is only looking at genomics, not using transcriptomics, which is always very vital for this kind of work. But this is locus of heat resistance. We did a query using these certain genes for locus of heat resistance of all our assemblies that we've had over the past year or 2 years. And we ultimately found around 11 salmonella isolates of varying serotypes that did have this actual locus of heat resistance. So this is a pathway that we're interested in going into. What else can we find from this data? Heat resistance, acid resistance, any of those type of intrinsic components. So in conclusion, we are focused. We've definitely invested a lot as an agency in moving forward with this technology and working with you all as our partners. We've built sufficient capacity. We feel we're well beyond the just capacity building point at this time. We're going to moving forward, what else can we do and add? And we want to continue engaging with our national and international partners. We have a lot of presence in both national and international meetings, including GMI. And finally, we want to use WGS, as we always have with PFG, use it in conjunction with all the epidemiological information that we have and bring in that totality of evidence to any type of investigation. With that, I'd like to thank all the people in the room, and thank you for all the people on the slide. A lot of the collaborations, I know for us, just standing up the program since 2014 took a lot of collaborations with everyone here and some of the people that aren't here that were here earlier in the week for the NARMS meeting. So I'd like to thank everyone.
Well, good afternoon, everybody. My name is Chris Braden. I'm from the Centers for Disease Control. Excuse me. I'm going to be talking a bit about how the different agencies that are implementing this new technology are actually coordinating. I think this is an important component of what we do. So an approach we've taken in order to establish this interagency coordination is really to build upon a history of collaborative programs that we've had. And some of you here may know of or have been involved with the Interagency Food Safety Analytics collaboration that was established before this particular collaboration and really builds on that type of structure. Of course, we want to apply advances in technologies, and the one that we've been concentrating on is next generation sequencing, but we want to scan the horizon to see what opportunities continue to evolve in the technology fields. Of course, even within next generation sequencing, as Martin had said before, there's going to be the next generation sequencing and how that's going to change what we do. We want to leverage the knowledge, expertise, and data among agencies. We bring certain levels of expertise in different areas together, and that makes us stronger, and certainly bringing our data together makes what we do more effective. And then set up a structure for our collaboration that is efficient and guided by strategy, and prioritize communications and stakeholder input over time. So the Interagency Collaboration on Genomics for Food and Feed Safety, or GenFS, was established in 2015 to strengthen the federal collaboration on the use of whole genome sequencing in food-borne pathogen analysis and investigation. Multiple federal agencies are involved, and most recently we've had ARS and the Animal and Plant Health Inspection Service, APHIS, at USDA join this collaboration. So really, GenFS is meant to support the implementation of a shared vision of coordinated networks for genomic sequencing. We want to use flexible tools and analyses and communications needed by the respective agencies to harmonize procedures and standards where we should, and really these two large networks are the ones that we've concentrated on making sure that we are collaborating and harmonizing as much as we can. So we have some targets for what we are prioritizing for our development, coordination, and harmonization, including some of the things that we've already talked about, the system tools and pipelines and methods, the analytic procedures and protocols, standards, sharing data and data availability, harmonizing some of the ways that we manage the networks with proficiency testing and training, how we use this data in surveillance, investigation, and research, and then the external communications and partnerships. So we have a draft charter that is actually undergoing the process of having our agency heads sign it as we speak. We have a steering committee with representation from each agency, and then we've set up these 4 work groups. One, data standards, analytics, comparisons, interpretation, and I'll show you some of the output from some of our work, interagency training, the network workflow harmonization, and then communications. So one of the things that we've had as a strategy and priority from the beginning is really sharing our work, our structure, and our strategy. And one of the things that we've done from the beginning is work to make sure that the DNA sequence and metadata that we produce are publicly available. So the data, as you've heard, is uploaded to the to NCBI and GenBank. That includes all organisms undergoing whole genome sequencing in PulseNet and Genome Tracker. There are clinical, food, and environmental isolates, and we must do so with the protection of personal and commercial information. We also make the tools that we use publicly available, either as open source or commercially available software, and then publish our methods and validation analyses. So the standards and validation, what that work group has really accomplished is establish the quality standards that are monitored for all submissions of whole genome sequence to GenBank, and to then develop and publish benchmark data sets, which I'll show you a little bit more about next slide. We'll do the validation studies. Some have already been mentioned, having to do with SNP. There's more to be done, I think, including when we validate the whole genome MLST, and really do some of the more careful cross comparisons of MLST and SNP analyses in individual pipelines, and then that cross validation, and then AMR genotype phenotype comparison. You've heard quite a lot about already, but there is another publication out of this work group that's pending. So these benchmark data sets, I think, we were just talking before, is I think a great resource for anybody. Certainly it's what we need internally to be able to validate a number of our analyses and pipelines, but being able to then provide them publicly allows those data sets to be used in validation and research studies for anybody. So we have 5 DNA sequence data sets consisting of 10 to 31 well-characterized outbreak and unrelated isolates that have been developed so far for Listeria, Campylobacter, E. coli, and Salmonella. And with each of those, there is publications that detail the outbreaks that they're associated with. And these data sets are available for download at this GitHub site. So we are making those publicly available. Harmonization across the networks, of course, this is important because a lot of the laboratories are both genome tracker and PulseNet laboratories, and they can't be doing 2 procedures for the same purpose. We've really tried to harmonize our procedures for the laboratories that participate. So for the training, the training is provided for public health and regulatory program partners in PulseNet and Genome Tracker networks. They're both CDC and FDA-sponsored courses. Staff from each agency participates in the training as training faculty, and training certification applies to both networks. So I think we've done a lot to integrate our training. And the same is true for standing operating procedures in the laboratory for whole genome sequencing procedures, for sample and library preparation, for sequencing procedure itself, for the data management and upload to NCBI, and then incorporating new and changing technologies, because for those of you to do this, you know that new chemistries come out periodically and they need their own SOPs to change accordingly. And then the proficiency testing, so the same proficiency panels, the same analysis, the same reporting is used for both networks. So communications, we've really tried to be able to communicate what is happening in the networks, what's happening, how the data is being used in surveillance, investigation, and regulatory functions. We have some industry collaborative forums. For instance, the Institute for Food Safety and Health has had a couple of meetings, and we've also had some forums in collaboration with the University of Georgia Center for Food Safety. Many presentations and discussions at food safety and scientific conferences, and you'll see some listed there. And then we are putting together a white paper for publication on the use of whole genome sequencing in food safety. So looking to the future, and this is what I'm really concentrating on, how we're going to be using this information on a day-to-day basis in our agencies. Whole genome sequence technology will replace traditional methods for routine microbiologic characterization of foodborne pathogens for use in surveillance, investigation, and agency action. Now, people ask, is still important to do some traditional microbiology because that gives you still additional information. And yes, there are times when that's going to be appropriate. But for routine for our routine purposes, we think that whole genome sequencing will give us the information that we need. Of course, to use these tools in this way, they have to be validated with comparable results no matter where the testing is done. We want to have shared tools, standards, and data for all stakeholders so that public health regulatory partners can use these standard tools, but in addition, those in industry, academia, and our international partners can also participate in using some of these same tools in order to be able to compare data among partners. And then the other thing that we're really trying to do is come up with some tools and methods that will be simplified for WGS analysis, because to do the research and to develop these does take a lot of computing power, and it takes a lot of bioinformatics expertise. Not every health department or other institution is going to have that kind of computing power and expertise, and so we do need to develop the tools that are more simplified in order to have many more participants in these networks be able to participate. So in those cases, there would not be a requirement for high-performance computing and advanced bioinformatics expertise in order to use these tools. And I would like to thank the genomics and food safety members, and especially the communications work group that's worked on this and other presentations, USDA FSIS for hosting this meeting that is meant as a meeting for all the GenFS members, and your interest and input. Thank you very much.
Thank you, Dr. Brighton. So now we'll have a question-answer session. So if all of the speakers from this federal-state collaboration session can come up to the stage, and they can all have the microphone. And if I could ask the panelists to just state your name when you answer the question for our folks online. Okay, we'll start with a question in the room.
Great, thank you. Betsy Barine, OFW Law. First of all, thank you all for your time today. I found the presentations fascinating, and as I've been sitting here and we were discussing some at break, sort of the totality of what's been presented, and I have a question, and I'm not sure who's the best person to ask it, and it may be coming from a place of ignorance. But as I'm looking and hearing what is being talked about, the number of sequences that are being, or the number of isolates that have been sequenced, one of the questions that I have in my head is what, and perhaps it should be focused on Genome Tracker, of the pie of isolates that has been sequenced, how much of that is clinical samples, how much of that is other isolate sources, and how much of that is regulatory? Because as someone who represents the food industry, I'm focused on the regulatory and trying to better understand if I see a slide that says papayas or meat and poultry have a high sense, what does that mean in the whole scheme of things? And so what I'm trying to get at is for industry to look at trying to do research or other groups in that regulated area, how much of those are regulatory surveillance, how much of those are for cause. And as I talk with my industry partners, better understanding some of that information may inform research decisions that can help when they say, why is an industry adding to the database? We want it to be targeted and want to understand what's going on in those isolates. I'm not sure who's got the right answer, if that's, again, maybe there's a report out there that I haven't seen, but I think it's really important as when we've done other risk assessment type of work, certain agencies, certain industries have an abundance of data compared to others, and does that mean there's a true risk there? What does that data tell us? So I'd be curious in your thoughts as we try to, particularly from an industry standpoint, better understand what that data means.
Thank you for your question. Yeah, my name is Chris Braden from CDC. I think you have to be careful in this database, I think, to know that this is not a statistically representative sampling scheme. It does contain, especially I think on the food and environmental side, those samples were collected for lots of different reasons, and actually I'm not sure that we have the data break out what's sampling for cause and what's sampling for according to some assignment or routine sampling that might be done. So that I think is one thing that you need to understand when you're analyzing that as a limitation to the data. There are more food and environmental isolates than there are case isolates. I think there always will be because food and environmental isolates just depend on how much you test, and there's only so many cases. So that's going to be the case going forward. But nonetheless, even with those limitations, I think that there's lots that one can learn about the breadth of genomic variation in any number of ways in these databases that is helpful to answer some questions, but won't be able to say that what to break it down by, for instance, sampling for cause or surveillance.
This is Glenn Tillman from FSIS as well. To follow up, one of Dr. Goldman's slides had that we've sequenced around 11,000 uploads into NCBI since inception. Around half of those are part of our non-regulatory SQL program, and those have been kind of moved to a different bio project, which could provide some opportunity to look at them, those strictly within that bio project. And one of my slides had all the ones that we contribute to, and they were actually called the NARM SQL. So the other 5,500 were collected as part of a regulatory initiative. So we do have kind of a half mix there. And again, the NARM SQL are moved into their own bio projects. So that'd be a good place to start, understanding what each one of those bio projects on NCBI might have within it.
John Besser from CDC. It hasn't come up today, but there's actually something called Voluntary Net at University of Georgia, which could be used by industry to anonymize the sequence data. And actually, it's a firewall between public health and Voluntary Net. So it can be used to compare the food and environmental isolates submitted by industry to clinical cases to assess risk. But we can't, we, the CDC, FDA, USDA, can't specifically query that database. We can ask them if there's matches. They can then ask their membership as to whether or not whoever submitted it would be interested in sharing that data. But the idea is that there's concern in industry that the information would be used in a punitive manner. And voluntary net is a safe place to submit data that can be used for risk assessments. And it's not exactly answering your question, but it's on a related topic.
Jorgen? My name is Jørgen Slund. I'm from Singapore. I love saying that I'm from Singapore. Okay. Especially with a Danish accent. But it's fantastic to be in the U.S. discussing something like next generation sequencing, because this is clearly an area where U.S. is leading. I understand that in some other areas, U.S. is stepping back from leading, but actually in this area, you are really leading. We don't need to discuss the integration between the different agencies in U.S. Maybe it's a good thing that you have many different agencies, because then you can move in different speed, and we have seen that also today. But I have one question in relation to maybe unifying the forces, not only within the U.S., but also with the rest of the world. So I was looking at resistance, and I understand that CVM is moving forward with a resistome tracker, and I understand that NCBI is moving forward with something else with a very long name that I didn't really get. And I understand that FSIS is using the WHO Collaborating Center thing from Denmark, the ResFinder. Wouldn't it make sense if there was sort of a concerted effort to try to produce consistent, uniform methodology and also tools, so that the rest of the world could really get a benefit out of U.S. leading in this area?
Thanks, Jürgen, for the question, and it's a topic we discuss quite a lot. And the need for harmonization in allele calls is essential. I mean, if we're going to take seriously antibiotic resistance as a global challenge that must be addressed globally, we need harmonized methods to have a common language. So I think that the discrepancies are being worked through between, say, ResFinder and NCBI, and Bill can tell us the latest on that. But I think just in general, it's essential that it be done so the outputs are the same. There might be tools with different interfaces that work with bionumerics, for example, or different types of analytics, but as long as the output is either the same or can readily be translated into a common language, then we'll meet an important objective. But I do think that it's essential.
So, Jorgen, we were just at the ASM Genomics Conference, and we had a roundtable with NCBI, the CARD database in Canada, and Ole from DTU was there. Although he's technically not ResFinder, he at least is Danish, which is close enough. And we agreed that we would at least discuss harmonizing our curation efforts, because the content in the database should be the same. The methods to apply them, I think, will have to follow after that, because we'll have to do comparisons to see if we get similar answers, and the extent of application of related sequences is something we need to look at. But yes, I would agree with what you're saying.
I appreciate your Danish honesty. That's great. I agree with everything that's been said, that we're moving towards that. I think what Chris Braden presented on the data quality is the really key harmonization point. If we share data quality, we can explore these different mechanisms for allele database curation for different systems. And as with any new technology, there has to be sort of a creative period. And I think we're in that now. But I sense, and so no one system has emerged yet as the winner. But as long as we share this core of data quality, I feel that we're all moving towards global standardization and curation. We haven't gotten there yet, but I think we all agree that that's the direction we're moving.
And this is Chris Braden again, and you raised the issue having to do with resistance determinants databases, but I think the same can be said for other allele databases, that we all should be coordinating in the types of databases we use, even if the management is distributed in some way.
Yeah, thanks, Chris. At least at CDC, we're envisioning the global curation of these allele databases for subtyping purposes. I mean, that's a really big issue, but the Institute, the Pasteur Institute in France has done curation for Salmonella serotyping for forever. We're envisioning a global collaborative curation of some sort, but I think it has to happen on a global level ultimately.
We have a question from the web.
The question from the online group is, do you worry about the education training that may be required so that people can adequately interpret WGS data findings?
So this is Chris Braden. Is the question, is this a priority of some, could you repeat, please?
Sure. The question is, do you worry about the education training that may be required so that people can adequately interpret the WGS data findings?
So yes, we do worry about that. It is difficult. Not only is it difficult to learn some of the bioinformatics that may be necessary to do this type of analysis in order to appropriately interpret it in a laboratory, but there's a whole other realm of disciplines out there that also need some training on how to interpret this data, and so probably the next outreach is with the epidemiologists, really to do the genomics for epidemiologists type of training, and there are a number of courses, and I know Martin Whedon is here before, and he's led some of those courses. And so that certainly is the case. But I think as we use this more, it's going to be, there's going to have to be more training with more disciplines for instance, in the regulatory agencies or with risk assessors and so forth.
So Dave Bakshu from the Minnesota Department of Health. So from a public health lab standpoint, we have additional challenges. We're a smaller group, and traditionally we've had experts in microbiology, identification, that sort of things. Many of our staff simply don't have the background in sequencing or have the understanding of how to interpret sequencing and how it works. We've been able to hire a number of new people that have that background, but trying to bridge that knowledge gap between traditional subject matter experts and then this new generation of sequencing group is a real challenge, and we have to work at it constantly to try to bridge that gap. And I agree with Chris that working out, educating our epidemiologists is also really a vital thing, so that everyone is always on the same page.
I just want to add one quick thing. This is Glenn Tillman. So a lot of the tools have come a long way since the 2015 ASM Genomic Pipeline meeting in Washington. A lot of the online tools have come about and really moved things forward. Bionumerics and with applied math has moved forward and really made things a lot more streamlined, allowing users to be able to really use that stuff. So I think that training is a very big part. I think the development of these newer analytical tools in more of a web-based form and or just any type of GUI-based form have been really beneficial.
Mark Allard, FDA. Once again, I'm going to do a comment and then another question. I just want to say that many people in the room are actively involved in international and global training as well, writing white paper documents on the why you should adopt whole genome sequencing and the specific case studies that they're best used. And so this is collaborations with the World Health Organization, Food and Agriculture Organization, and the OIE. And so there's ongoing efforts for global trading because of global trade in food. So my question is, it's a little off the topic while we're here at USDA, but I know that the Department of Health for New York, they don't just sequence the 4 or 5 foodborne pathogens, that they're actually responsible and having sequencing efforts in almost 20 human pathogens. And so this question, I think, starts with Dave Boxford at Minnesota is, do you see a similar expansion of sequencing for other species? And what do you see as growth? And I guess then the question is, are you seeing equivalent support from your federal partners, both in databasing, et cetera?
Yeah, it's a great question. But PulseNet and foodborne is definitely in many ways the leader in sequencing. They're kind of the bull because there's so much of it. But we are involved with a lot of different types of pathogen testing, Legionella, Strep pneumo, MRSA. I believe it's about 10 different projects that we're involved with. And many of these we've reached out with our collaborators at CDC and said, we're interested in this organism. We have background in this. Let's work together. And they've been very, very willing to do that. And that's been really key for us. One of the real challenges with using sequencing with foodborne disease surveillance is while it's an awesome technique, it's an awesome method, if you batch it and do it once a month, you're really slowing down the process. And so we fill in our runs with other organisms, with other pathogens. The Legionella group from CDC has a really nice partnership, and they've created I think there's about 6 laboratories that are doing a study with them. The TB group is doing, from CDC, I believe they funded one site to do essentially all or most of the clinical TB cases throughout the country. There's a flu group, we're not involved with this, but there's 3 sites that are doing a lot of influenza testing. So there is a lot going on, but some of the public health labs are not always embracing the new technology for different organisms. So I think hopefully as sequencing becomes more routine at all public health laboratories, that they will consider doing other pathogens, because I do think this is a technology change that is not going to go away, and it's going to continue to provide additional important information for different pathogens.
Question from the room.
Martin Wieben, Cornell University. So we talked a lot about standardization of databases, allele calls, quality control. Where are we with regard to standardization of metadata? And I'm going to start out with just even food data. Yeah, I think porcine, pork, different names call the same thing. If we then move to environmental, data, to how do you describe an environmental sample, it gets even more challenging. And I think there's an importance to it that hasn't been mentioned. I think when we try to interpret SNP differences, it obviously depends on the environment an organism is in. If a salmonella is in a dry environment, it might only replicate 10 times a year, and therefore accumulate SNPs at a much, much slower rate than if the same salmonella sits in a poultry house, where it might replicate every hour because it's actively infecting one chicken after the other. And so if you don't have that information, some sort of standardized way, we sometimes will run into challenges how we interpret SNP differences. So is there a parallel effort? Is it part of FSGen to also standardize those data, or is that sort of a next step?
I can only tell you about the things I know, and that is at the Global Microbial Identifier and part of an ISO working group on next generation sequencing for food safety, there's an effort to use some metadata ontologies to standardize. This is called GenEPO. It's been worked on by the Canadians. CIFSAN has said that they will look at using that in their metadata. But I should tell you that what we would rely on then is probably the submitters to apply those terms in a standard way, because a lot of people will say that ontologies are the solution to everything. They're not if they're not applied in a standardized way by all the people contributing. Since we are the place where we're integrating all the data, if everyone in the U.S. goes a certain way and then the Europeans do a different thing, we're sort of left at the mercy of that, where we would have to either invest time and effort on our side to standardize the metadata once it gets integrated and submitted to us, or we just leave it alone and only worry about the standardized metadata in the U.S. So I know someone from CISAN could probably mention that they're looking at this. You could probably talk to them. I don't know if USDA is doing that. They probably will. You guys could probably talk about this.
We got a question from the web.
Yeah, Chris Braden again. We started by trying to standardize what the metadata fields would be in the GenFS agencies, and so it as a place to start. But I think it definitely needs to expand and to be able to have all partners be able to participate with some standard expectations.
Okay, we have a question from the web.
Can you say more about providing safe harbor for producers, manufacturers, others in the industry to A, gain familiarity with WGS, and B, provide useful information to regulators without bringing regulatory response down on their heads?
I'm not sure we have. This is Chris Braden. I'm not sure we have the representation here, but I don't think I really understood the question. If you could ask again.
Sure, I can repeat the question. Can you say more about providing safe harbor for producers, manufacturers, and others in the industry? A, to gain familiarity with WGS, and B, to provide useful information to regulators without bringing regulatory response down on their heads.
So yeah, safe harbor. I think that's the where is that safe place to be able to start to implement this kind of technology in order to learn more about your particular producing environment if you're in the industry, for instance. I think it is an important point. We have had a number of discussions with industry members about what might work if there's a third party that could be responsible for holding the key, for instance, and not releasing any of the identifiable information and data sets. I would hope that we can find such partnerships to be able to do so. John Besser had mentioned one at the University of Georgia called Voluntary Net, which is a industry collaboration. I know that at the Institute for Food Safety and Health, they are also considering being able to provide that kind of third-party resource. There may be other resources out there, and I think, for instance, IEH has said that they can certainly provide the service and provide that kind of data back that would be helpful to industry members to understand how to use this information. So I think there are some resources out there. It hasn't been widespread to my knowledge. Maybe IEH is maybe used the most, but I think it's certainly worth exploring for industry members. Is Mansour still here?
Mark Allard, FDA. I can comment just a little bit about this. We've been doing outreach to industry. FDA primarily works with the Institute for Food Safety and Health, IFISH, which is out at the Illinois Institute of Technology and our Moffitt Center in Chicago. And essentially, from 3 or 4 years of speaking to food industry, probably the most feasible path to learn new things about this technology is to work with a third-party provider and essentially do this online. Steve Musser touched on this earlier today. There's many companies that are involved in this, as well as many academic folks like Andy Benson in University of Nebraska and Martin Weidman at Cornell. But there's companies like Eurofins, IEH, Ecolabs, NSF International. You can find these on the internet that provide genomic services and can assist you. This is an expanding market because there are a lot of people in industry that would prefer to do it as a third-party provider as opposed to building their own laboratory. So we can give you more information or reach out to your local lobbying group, the GMA, United Fresh, SQF, IFAP. There's many speakers at these meetings that have a diversity of services to the food industry.
There's a question from the web.
There was no mention in today's talks by the different agencies on how WGS is being used in the detection of outbreaks and for carrying out outbreak investigations. How is the CDC using the information, and how is the integration with what the FDA, FSIS might have in their database taking place?
So as I mentioned in my presentation, basically we will be transitioning all of our traditional characterization techniques over to whole genome sequencing. And so whole genome sequencing will be replacing pulse field gel electrophoresis in our cluster detection, and it will be replacing the assays that we use for isolate confirmation, serotyping, virulence typing, and resistance typing. So we will be, all those separate assays will now be carried out with one assay using whole genome sequencing. In the future, we're in the midst of that transition now. As David Boxrud had said, that it's a difficult time because we're actually doing both traditional typing and whole genome sequencing, and that makes it more expensive and more time-consuming. But we anticipate that in the long run, this will be time and cost saving. So that's how we're looking at moving forward, and I think that I can talk for the other agencies to say that that's their plan too. As far as how we use this data together, well, we do talk a little bit about Genome Tracker database and PulseNet database and so forth, but in reality, we're all submitting all of the data into a single database, and we're just contributors. So PulseNet is contributing to a single database, Genome Tracker is contributing to a single database, and we're all using all of that data in our analyses for outbreak detection, investigation, surveillance, and action. So that's how it comes together.
I have a question. So John mentioned that by the end of fiscal 18, I believe that there would be all or close to all salmonella would be sequenced, and I'm just curious about any logistical issues that would be raised by that, and what do you expect at the end of fiscal 18 when we have all these new salmonella sequenced in real time?
I'm John Besser. That's a very good question, Peter. Yes, we definitely, it's not going to be smooth sailing. We're really getting into a whole big data era. I think the experience of Public Health England would probably be helpful. They saw a dramatic increase in the number of clusters that needed to be investigated. We saw this with our Listeria Combined Initiative as well. So I think there's going to be a need to develop new tools for cluster triage. What clusters are most likely to be productive? I think there's going to be more There already are more clusters to investigate than there are investigators to investigate them. Sorry for the convoluted sentence there. But I think this is going to become a major issue, and the investigative resources are going to be a problem. I think we're working to streamline on the laboratory side to make these things easier. For instance, you heard about nomenclature that's being developed, which will allow for ready recognition, easier recognition of clusters, and easier communication. But I think a lot more work needs to be done in order to prepare for this big data era that's rapidly approaching.
There's a question in the room.
Yep. Sherry McGarry, CBC, and the question is mostly for Dave at Minnesota Department of Health, but I'd welcome other panelists to weigh in. And it piggybacks on what you're just talking about. So when PFG first came on board, it took longer than it takes now, right? So there are efficiencies that were gained and techniques that were modified to make it faster. Where do you see some innovation at the state level to kind of speed things up a little bit? I know we're still at the early phase, but maybe this is also an innovative phase too. So where do you see efficiencies to reduce the total amount of time?
Thanks, Dave Bakshi from Minnesota. Yeah, there's a lot of ways to make the process more efficient, and that's something as we continue to do more sequencing, we're going to have to continue to look at these ways. We're going to have to adapt to new technologies. You know, the work that we're doing with Illumina MiSeq, 5 years from now, we may have a completely different technology than what we have. For us, as we're really ramping up the amount of sequencing that we're doing, it's a lot of training, a lot of getting everyone on the same page within our laboratory. And as we're doing that, we're okay with a little bit of extra time to make sure that our quality is correct and that we don't have any issues. Once we really get this incorporated into a standardized workflow, then we're going to see what we can do to try to make it more efficient. We're in the process of getting a library prep instrument that will take some of the time and the labor of creating the library prep, which is one of the more labor-intensive parts of the process. And also the kits that we're using, the technology is changing. The MinION is could be a faster tool than what we have right now. And right now, for PulseNet and various other things, it's probably not ready for prime time, but very, very soon it will be. So I think all of us are going to have to continue to adapt. But what is great about sequencing is if you have that quality of sequence, if you use a new technology, they're very comparable going forward.
That's a good answer, Dave. This is John Besser, and I think Dave already mentioned the batching issue, which is a big potential problem in turnaround time. But as he mentioned, there's also new technological developments. A new library prep and DNA prep chemistry just came out recently, and we'll be looking at all of those. But I think what I didn't have time to talk about earlier today was some of the new tests. You asked about innovation. One in particular, amplicon sequencing, we're looking at a quasi-whole genome MLST that can be done directly from a specimen. And that actually could shave weeks off of the whole process if successful, but it's still highly experimental at this stage. But I think ultimately where we want to go towards is a direct from specimen testing, which will have the biggest overall impact on turnaround time in terms of, because the culture is the slowest part. Often culture happens at clinical sides at the state and sometimes even at the CDC, and that can really slow down the whole process, and we're hoping to bypass that at some point in the near future.
We have a question in the room.
Yeah, David Goldman, FSIS. I just wanted to go back to the question that was asked online about outbreak investigation. And when the question was asked, I realized we really hadn't done a recent case study of an outbreak where whole genome sequencing was used. And I think, everyone knows we rode PulseNet with PFGE very successfully, highly successfully for 20 years. And, if two patterns were indistinguishable, we said, okay, everyone agreed with that. We even dealt with one-band differences very well, I think. But now with whole genome sequencing, the picture is less clear, as you've heard many speakers attest earlier. And I want to go back to something and emphasize something Martin Weidman said earlier, which is that, to me, where we are now is that the epi is more important than it was using pulse field analysis as a way of judging and ultimately including cases in the case definition. I'll just briefly reference a very recent outbreak in which we used both PFGE and whole genome sequencing. And in this particular instance, the PFGE was done first by most of the state labs who were members of this outbreak, affected states. And what we found out was that after the PFGE seemed to include cases, a week or so later we'd get this genomic sequence information which would exclude those cases. So there was this sort of sequential activity which proved quite challenging for us as a federal family with the state partners in trying to determine whether this outbreak was over, have we fallen below the epidemic threshold, or are we still above it? And so this was a very recent example, and I think while we're in this transition phase still using both tools, PFGE and whole genome sequencing, we may find similar challenges going forward.
All right, Joe Heinzelin from Neogen. One of the things I had hoped would be addressed in some of the talks today, and maybe it will be tomorrow, is around the question of the statute of limitations around isolates in a database. Specifically, let's say you find an isolate in a facility, you take corrective actions. Does that absolve you from that type of isolate in the database? Are there things that people can do or use this data? So I guess what I'm really trying to get to is, what can people do with whole genome sequencing data once an isolate is in the database from a facility to say that they've made corrective actions or they've used this technology? And how long is that data point still real and applicable throughout the life of the food?
Yeah, so I'm not sure if we have somebody in the regulatory community that can address that.
You're always asking sort of policy-related questions is always the third rail of the FDA. But I'll just make a comment. And so the emphasis is that a genetic match is not a regulatory decision. A genetic match is an indication of a shared common ancestry, shared isolates, and it's a presumed link, a potential link to a food and clinical or whatever gets matched. But inspection and epidemiology have to follow, and in fact, FDA will not do any regulatory action on genetic match alone. There must be an investigation. So we've seen 5-year-old matches, 10-year-old matches. Essentially, this leads us back to a facility, to a region, a country, a state, but it depends what the investigator finds, and it depends how the company responds. There's a whole process that hasn't changed. We just have a new genetic tool that helps establish linkage. And so if investigators go and inspect a facility and it's clean as a whistle, then there's nothing to be done. There's no regulatory activity. If the inspectors come and they found positives from foodborne pathogens, then it's like what Dr. Musser said. It depends on whether there's an association with clinical or not. And so it depends. But the databases last forever, or at least 30 years. I know I have data in the database that's been there for 30 years. So our full expectation is NCBI will not go away anytime soon, and that we'll continue to see and use these linkages.
This is John Besser. Mark, thank you for that response. I think that was an excellent response. I just wanted to suggest that we might, because this technology is new, we're suddenly able, with more specificity, to connect current cases with past isolates in the database, that this may be less of a problem in the future because we'll be able to connect cases more or less in real time with isolates as they're being found in the environment. I think this is a circumstance where we've got the new meeting the old, and I think it may not be as much of a problem as people are concerned about. And as Mark pointed out, this activity is not new. We've always compared to the historical database. What's new is the specificity with which we can make that connection.
Okay, so this is a last chance for questions on the web, in the room. And seeing no questions, we're going to start up again tomorrow morning at 8 a.m. The first set of presentations will be on what's happening with our foreign international organizations, Mexico, Canada, and also globally. And then we'll have some presentations by our stakeholders, the food industry and other stakeholders. So really looking forward to the presentations tomorrow, and I hope to see everybody here. And again, we'll be starting at 8 a.m. You'll enter through the fifth wing, and if there are no other additions, then we'll break until tomorrow. Thank you.